Thanks so much. I'm really happy to be here. So today I'll talk about prenatal PFAS and reproductive outcomes, mostly birth outcomes in the ECHO study, which some of you may be familiar with. And then I also will save a few minutes to also share some of my work that I did during a COVID extended sabbatical in Germany that looked at prenatal PFAS in relation to vaccine effectiveness and infection. So hopefully it can follow up nicely with Andre's work. So for those of you who are not familiar with the ECHO study, this is a consortium or a program of prenatal cohorts across the US and in Puerto Rico as well, and Hawaii. I guess that's still inclusive of the US. And so we were interested in looking at birth outcomes in relation to per and polyfluoroalkyl substances and maternal stress across all of these different sites in the US. It included over 50,000 kind of maternal child pairs and 50 cohorts. For our study, we included 11 cohorts that had PFAS concentrations available during pregnancy. This is a list of them in case you're familiar with any prenatal cohorts across the country. And the birth outcomes that we were interested are related. Whoa, this is going on its own. Maybe I'll just keep an eye on that. So related to birth weight and gestational age. So we were concerned about birth size, which included birth weight for gestational age Z-scores, small for gestational age, which is in the smallest 10th percentile, and then large for gestational age, and then term low birth weight. And then we also were interested in preterm birth, which is a kind of distinct but somewhat related outcome, just looking at the duration of pregnancy with less than 37 weeks being considered preterm. So I'll show you the results of all of these outcomes, but just to keep in mind that they're all quite related. So even though we looked at multiple, they're not distinct necessarily. So we measured 12 PFAS in these prenatal samples. So some of the cohorts actually that just had serum or plasma available but hadn't measured PFAS were able to join this group, and so we measured additional ones at the Wadsworth lab. And the total study population included over 3,000 participants over a 20-year range. So from around 1999 to 2019, and as I mentioned, in 11 different prenatal cohorts. So 5 of the prenatal PFAS were detected in over 60% of the samples, so we focused on these 5 for analyses. We adjusted for maternal age, education, race, ethnicity, and parity, which is kind of speaking somewhat to the breastfeeding, potential confounding with breastfeeding. And then, it's still going on its own. And then looked at stress, maternal stress as a modifier using the perceived stress scale. In 8 of the cohorts. We also looked at a mixture of PFAS using Bayesian weighted sums. We also examined, because of these prenatal cohorts kind of existed before they all became a part of ECHO, PFAS were sometimes measured at different trimesters, so we were interested to know if the trimester it mattered when the PFAS was measured during the pregnancy. And we also looked at infant sex as a potential modifier. So overall, we found that PFAS were associated with smaller babies and babies born too soon. And you'll see that the robustness of the results maybe varies by outcome, but this is kind of the overall message. We did not find that maternal stress in this study modified the associations between PFAS and birth outcomes. So where often we do find that this kind of double jeopardy of being exposed to environmental stressors and social stressors does have more outcomes. In this case, it didn't. And then we did find that associations were stronger among male infants in some cases. So we did look at these both continuously and by quartile. I'm going to show the quartile analyses because I think it's easier to digest more quickly. So this shows for each of these PFAS the three highest quartiles compared to the lowest quartile. And you can see that, so with increasing levels of PFAS, we found lower birth weight or for gestational age Z-scores, which means smaller babies. So, and as you can see across these quartiles, there is, evidence of an exposure response with shown here, and PFNA and PFDA were statistically significant among comparing the highest to lowest quartile. So here are the continuous results when we looked at birth weight for gestational age Z-scores with each of these PFAS. As you can tell, these is much harder to digest, I think. But just to kind of show what the numbers looked like and to show it stratified by infant sex, that there's decreases in birth weight for gestational age Z-scores. And so for PFOA and PFOS, we did find stronger associations, but really it was the same story for all of these estimates. So when we looked at large for gestational age, you see kind of a similar pattern somewhat, and PFNA and PFDA stand out. So, but this is showing a reduced risk of large for gestational age. So large for gestational age and small for gestational age are both adverse birth outcomes. So this is kind of a Goldilocks outcome. We don't want too big or too small. We want them right in the middle. However, so finding reduced risk you'd think is a good thing, but which is true, but it's also, again, in this, if you think of the whole population distribution, that it is shifting it to towards smaller infants. But this is somewhat of a sort of provocative result. And then for small for gestational age, we're finding an increased, with growing quartiles, an increased risk, but none of these were statistically significant, at least across the quartiles for small for gestational age. So the shift really seems to be on the other end of the spectrum. When we looked at term low birth weight, again, these are all births that are all born the full term, so after 37 weeks, but were smaller than, that were less than 2,500 grams. And although PFOA and PFOS do stand out as statistically significant, you can see also by these error bars how imprecise maybe these estimates are. And then these are the results for gestational age. So this is kind of a more continuous look at preterm birth. None of these reached statistical significance with a quartile analysis. So this part was a little bit less clear when we chose the when we dichotomized it at preterm birth less than 37 weeks, then PFOA does stand out a little bit. So when we looked at the mixtures analysis and combined all the PFAS together, we did find pretty consistent results. So again, smaller birth weight for gestational age, increased risk of small for gestational age, decreased risk for large for gestational age, and increased risk for term low birth weight. And then, although not statistically significant, we found an increased risk of preterm birth and shorter gestational age. One aspect of this analysis, too, is that it notes which of the PFAS contributes most to this mixture, and PFDA stood out in that respect. So these results are published in case anyone wants to dig deeper into them. This was also a huge group of people that contributed to this these findings. And overall, I think the implications show that I think this confirmed a lot of previous studies that had been somewhat equivocal in the literature, but it did confirm that findings of decreased birth weight associated with high prenatal PFAS, and this has been the largest study to date. There were no differences by trimester, which was of concern in previous studies that pregnancy hemodynamics and blood volume changes throughout pregnancy may impact results. So we were able to put that to rest. And then, and also that the study estimates PFAS mixtures, and that these findings kind of underlie the importance to ban all PFAS, which I think everyone is on board with here. And then, sorry, I feel like I'm wrestling with my slides right now. And that, and just that this gives an additional opportunity to show the impact of PFAS on public health outcomes. I think it's also interesting that the vaccine work kind of stands out in, when people are considering what sort of the toxicity or the public health impact, but birth outcomes kind of feel like get a little less importance. I don't know what to attribute that to exactly, but I'll show these other results as well. So just to shift gears, so to show this impact of maternal PFAS exposure on vaccine antibody levels and childhood infections. So this pregnancy cohort in Hamburg, Germany, followed pregnant people and their children. They had tons of visits and biospecimen collection and questionnaires throughout this entire study period, and they were really focused on the immune system. So they didn't care at all about PFAS, and were really focused on understanding the kind of crosstalk between maternal and child maternal and child immune systems. But they had data on vaccine antibody levels and really detailed infections. But they also had a lot of banked serum, which they kindly let me borrow or use. And I sent to a colleague in Sweden to calculate or to quantify the levels of PFAS. So just to kind of, these are pretty preliminary results. This work is not published, but these are the associations. Wait, I feel like there was another. Oh, I feel like there's a slide missing. So we looked at 18 PFAS. I know you wanted more than 6, so we've got 18 in relation to measles, mumps, and rubella, and Tdap, and the number of infections. So just to start with measles, mumps, and rubella, this is across all of the, so of the 18 we measured, I think actually I have here, I want to say 11, but 2 of them are both PFAS, different kinds measurements of PFAS, so I kind of consider it 10. They got combined in the mixtures analysis, but essentially the measles, mumps, and rubella, we did not see strong associations with the exception of P. funda and rubella. So we did find higher levels of P. funda associated with lower titer levels of rubella. And this is again comparing prenatal PFAS to vaccine titers at 5 years old in children. I think that was the slide that I was missing somehow. And then when we looked at the Tdap, so tetanus, diphtheria, and pertussis, we found increased levels of P-fexis associated with lower titers of tetanus, and same with diphtheria. And then when we looked at childhood infections, we did this, the parents were asked each year about the infections in the previous year. And they kept really detailed, they knew they were going to be asked this, so they kept detailed records of this. And so in the first 2 years, there really wasn't much of an association. And in many cases in Hamburg, Germany, children do stay home. Usually the first year and sometimes the 2 years before going to a kind of preschool-like daycare situation. So, but in year 3 is when you see the increase in infections with higher levels of PFNA, PFAS, and PFUNDA. And then year 4, we also see those increased associations. Year 5 kind of falls apart a little bit, but the total is also there. And this is just a QR code to these preliminary results since they're not published. So just want to finish by thanking everyone from the ECHO study, including Rachel, who's here. But this is a very long list of contributors, and then also the group in Hamburg and Europe. So I'm happy to take questions at the end.